So in part 3, we're going to look at the nucleus. It is the largest organelle. It contains a genetic library of blueprints for the synthesis of nearly all your proteins. It does respond to signals that dictate the kinds of amounts of protein that do need to be made or synthesized. Now, most cells are going to be uninucleate, which means they have one nucleus, but some may be multinucleate, like skeletal muscle and certain bone cells like osteoclasts and some liver cells, while others may be anucleate, which means have no nucleus. So here is a red blood cell, which doesn't have a nucleus, and then there's some white cells that have a nucleus, and then here's one of those bone cells called an osteoclast, and you can see it has multiple nuclei in there. So in section 3.9, we're going to look at the structure of the nucleus, and your objective here is to outline the structure and function of the nuclear envelope, nucleolus, and chromatin. The nucleus has three main structures. We're going to have the nuclear envelope, nucleoli, which is plural for nucleolus, and the chromatin. So you can see here there's a nuclear envelope. It's actually a double bilayer. So there's an inner and outer membrane, and then the chromatin is how your DNA is contained within the nucleus. It'll have proteins associated with it, and then a dark region called the nucleolus. So looking at the nuclear envelope, it is a double membrane barrier, and it encloses the nucleoplasm, which is jelly-like fluid. The outer layer is going to be continuous with the rough ER, so the outer membrane of the nucleus folds outward to make the endoplasmic reticulum, and the rough ER, the rough endoplasmic reticulum, is going to be studded with ribosomes to make it look rough. We've seen that earlier in the chapter. The inner layer is going to be called the nuclear lamina, and it has a network mesh of proteins that help maintain the shape of the nucleus, and also acts as a organizing or scaffolding area for the DNA. There's also going to be nuclear pores, and these are going to allow substances to pass into and out of the nucleus, and they are guarded by the nuclear pore complex, which is some proteins there, and that'll regulate the transport of specific large molecules in and out of the nucleus. Over here we have a scanning electron micrograph showing the surface of a nucleus, looking at the outer membrane, and then if we zoom in, so higher magnification, you can see the nuclear pores. Just circle one right there. And then if we look at the nuclear lamina, which is the inner layer there, you can see this network mesh of proteins that helps provide shape and support for the nuclear envelope. The nucleoli, or nucleolus singular, is going to be a dark staining spherical body or bodies in the nucleus, and these are involved in making the ribosomes, or the RNA that makes that is part of the ribosome. It's called ribosomal RNA, and so that's where your ribosomes or your protein factories are made. Once they're made there, then they come out into the cytoplasm through the nuclear pores. Now, the nucleoli are also associated with the nucleolar organizer region and contain DNA that would code for our rRNA, which is ribosomal RNA. Now, there's usually only one or two per cell. And then we have the chromatin, and the chromatin is how your DNA is found within the nucleus. It consists of 30% DNA, 60% proteins called histones, and 10% RNA. And they're going to be arranged. There's going to be fundamental units arranged here called nucleosomes, and here your DNA would be wrapped around the histones. And you can see here's your DNA here at the top, your double helix, and then as we begin to condense the DNA, it's going to wrap around 8 histones, 2 turns for every histone, and then there's linker DNA. And here, if we need to access the genes within the DNA, the coding within the DNA, then we would need to unwind that DNA from the histones, so we can block the coding of DNA by altering those histones that are there we might add certain molecules to there that keeps those genes from being expressed. So that's what this part is talking about here. If you alter those histones, the alteration of those histones can regulate whether or not those genes are expressed. So you can regulate gene expression. It's kind of like on and off switch there. Now, chromosomes themselves are only going to appear during cell division, and here it's where the chromatin condenses further, it winds up further and further and gets thicker and thicker, and so we would refer to that as condensed chromatin, forming those chromosomes. And this is going to help protect your fragile chromatin during cell division. So it's like packaging it up. So you can see we wind it up, and each copy of our new DNA is going to be forming one of these sister chromatids here, and that would be your entire chromosome there during cell division. So you would only see chromosomes during cell division, and chromosome literally translated means stained or colored body. Chromos means color, and soma means body. So which of the following cell types does not possess a nucleus at maturity? Is it skeletal muscle cells, red blood cells, bone destruction cells, those osteoclasts, or liver cells? So this is part D of chapter 3 lecture notes on the cell, the living units. In this part, we're going to continue our study of the nucleus. And so in section 310, the cell cycle, your learning objectives are going to be to list the phases of the cell cycle and describe the key events of each phase, and to describe the process of DNA replication. So the cell cycle itself is a series of changes a cell undergoes from the time it is formed until it reproduces. There's going to be two major periods of the cycle. There is going to be interphase, and during this phase the cell grows and carries on its usual activities, and then the mitotic phase, where the cell divides into two new cells or two daughter cells. You can see in the diagram on the right, interphase visually on the diagram looks like it takes up most of the diagram, which kind of shows that that's where the cell spends most of its time during that cycle. And then the yellow area there on the bottom is the mitotic phase, where the cell goes through a division. In interphase, which is green on the diagram there on the right, this is going to be the period from cell formation to cell division, when it carries out most of its routine activities, and if it's going to divide, the cell would prepare for cell division during that time. During interphase, if you're looking at the cells under the microscope, you won't be able to see them because they'll be uncondensed as chromatin, and chromatin is too thin to be able to see with a light microscope. Interphase has some subphases. They include, and you can see them there, as G1, S, and G2. And it's during this time if a cell decides that it has the right stimulation to do so, it's gonna have to copy the DNA in itself so it can distribute to the two new cells, so that each cell gets all of the instructions necessary to run the activities of the cell. So the subphases are listed here. There is G1, which stands for gap 1, and during this phase, sometimes it's like they may say this is their growth going on here. Here the cell's going about growth and normal metabolism, it's going about its normal business. Some cells will stay in this stage permanently, like a nerve cell, and if so, we'll call it the G0 phase. But for those cells that do divide, if they make it to a certain point and the right activity in the cell is going to cause the cell to divide, then it enters the S phase, which stands for synthesis. And here is where copies of DNA are made. And then after the DNA is made, then we get into G2 or gap 2, and that's where we're going to be making final preparations for that cell division, because the cell has committed to that. So you see here, you have, let's start with these two new cells dividing from the prior cycle, and each cell would go through G1, growth, and then get into synthesis. If the cell passes that point, replicates the DNA, and then we have our preparation for division during G2. So DNA, in order for the cell to divide, we have to make copies of all the chromosomes. We have to make copies of every piece of DNA because it contains the instructions necessary for the cell. So we have to do this. Now, DNA itself we know is a double-stranded DNA helix, and in order to be able to replicate it, we're going to need an enzyme called helicase that is going to unwind and break apart those hydrogen bonds, so unwind and unzip. And when this process is occurring, the point where the helicase is opening up the DNA, where the strands separate, is called the replication fork. And the area of active replication is referred to as a replication bubble. And each side, when we pull the two strands of DNA apart, each side will form a pattern for making the new strand. So replication is going to start once helicase unwinds the DNA molecule, and both strands are exposed now, the bases are exposed. An enzyme called primase is going to start replication by laying a short strand of RNA, and we're going to call that short strand of RNA an RNA primer. So there are some other enzymes involved here because we're going to be making a polymer of DNA. It's the name of the enzyme is DNA polymerase, and this DNA polymerase will synthesize the new strand, and it's always going to add new nucleotides to the 3' end of the growing chain. So we can only go in one direction. So we're going to have DNA polymerase working on both strands, but one strand we're going to be able to build continuously as the DNA helicase unwinds the DNA strand. So let's say right here we have our DNA helicase. It's moving along the DNA double helix this way, and it's unwinding and unzipping the DNA. So right here would be your replication fork, as indicated there in the diagram there. And as we're moving to the left along the DNA strand, here's your DNA polymerase adding new nucleotides according to complementary pairing rules. But if you think about it, the other strand is going in the opposite direction. That is, the 3' end is on the other side, and DNA polymerase can only add to the 3' side, so we have to open up some DNA, set a primer down, and then have DNA polymerase work in the opposite direction. And so what we're going to end up having is segments being made as we open up more strand. Primase will go and lay another primer and then lay another segment down. So those segments there eventually need to be binded together, and that's done so by an enzyme called ligase. But going back up here, the strand that we're able to replicate continuously, which in this diagram is the top strand up here, that's going to be called the leading strand. And then the other one that we have to replicate in segments is called the lagging strand. Okay, so I found here's another diagram here that shows all the enzymes involved. We have our helicases here in the diagram, and our primer had already been set somewhere back over here when we started the replication. And so as the helicase moves along the strand, unwinding and unzipping it, we're adding new nucleotides to the 3' side. So this is on the new strand is the light blue color. So this is the 3' side over here, and we're able to just add continuously. But on the other side, we're constantly having to have primase lay down a new primer, which is in red, and then our DNA polymerase can come in and start building these fragments. You can see an old primer from earlier, and we're adding, going in the opposite direction. And once we start adding that segment, the segments are not bonded together, so that's where DNA ligase comes in and will bind each new segment that's formed. And this process continues until the replication stops. By the way, the same process is going in the opposite direction. So once we open up the DNA molecule, DNA is replicated in both directions. The thing is the side switch. As we're going in the opposite direction, the lagging strand and the leading strand are on the switch sides overall. So what is the end result? You're going to end up with two identical daughter DNA molecules, and they're formed from the original one. During our mitosis cell division or mitotic cell division, each new cell is going to get exact copies of the DNA that were made during the DNA replication event. And so we have 46 pieces of DNA. We got 23 from our mom and 23 from our dad, and so we have to make copies of each of those so that each new cell gets a full sets of chromosomes from both maternal and paternal. So each of the copies are going to be moved into the new cells, so that the cells get all of their genetic information and all the chromosomes. Now, because you start off with 2 old strands with their bases, I'm just going to draw a little basic double-stranded DNA. When we separate the two strands as they're open, they form the template for adding a new strand on either side. So when we're done, each new copy of DNA, exact copy, is going to be one old strand, and the other one is going to be a new strand. And the same is true for the other side. So once we finish replicating the whole thing, your 2 new copies of DNA are going to be one old strand, one new strand, and that's going to be referred to as semi-conservative replication, because semi means we've saved. Semi means half of it is saved, half of the original DNA molecule. So the conservative means to save half. That's the old strand. So that's the model to use. So now, once we've gotten through copying our DNA during replication, we've gotten past the S phase, we need to do those final preparations in G2. And eventually we're going to enter the M phase, or mitosis, the mitotic phase. And so most cells will do this. There are some cells that don't divide rarely or at all, and if they do, it's very, very slow process. Examples include skeletal, cardiac, muscle, and nerve cells. So if any damage is done to those tissues, it's not going to be repaired with new cells of that type of tissue. Instead, you would get just scar tissue or fibrous tissue there instead of the functional cells like a functioning nerve cell or a skeletal muscle cell. So the mitotic phase, or M phase, of the cycle would begin after we finish G2. And there's going to be 2 events, and they overlap somewhat: the mitosis and then cytokinesis. And we do have to have some control over this process. We don't want cells dividing out of control. So there are some control mechanisms in this, and they involve these checkpoints that you see here and here on the diagram. But you can see on the diagram here, during the M phase, or mitotic phase, we have mitosis, which includes these subphases: prophase, metaphase, anaphase, and then there's a telophase, and then cytokinesis. But you can see cytokinesis, as I stretch out, it goes back and it actually begins during anaphase. And cytokinesis is where the entire cell divides. Mitosis is just the nucleus, and within the nucleus, which is the chromosomes, the new copies of DNA being divided into two new nuclei. So mitosis can be thought of as division of the nucleus, and cytokinesis is division of the entire cell, all the cytoplasm, after the nucleus is divided. So we're going to look quite closely at the events of mitosis, which is again the division of the nucleus, where we're going to divide or separate our duplicated DNA, which are going to be condensed into chromosomes at this point. There's 4 main stages. PMAT is one way to think about it. P for prophase, M for metaphase, A for anaphase, and T for telophase. And so looking at the cells, we're going to actually divide prophase into early and late. So here's prophase. It can be broken into 2 parts. We have early prophase, and here your chromatin is going to condense. And at this point, when it condenses, you'd be able to see it with the light microscope, and so they're called chromosomes at that point. Each chromosome is going to have both duplicates of the DNA that was copied during the S phase. And each duplicate is still connected, and we're going to call each duplicate sister chromatin. So when we draw our chromosome, and the chromatin is real condensed now, and it's become thick and short after condensing it. Each one of those is going to be connected in the middle at the centromere. And each one of those is going to be called a sister chromatid. So here and here we have our sister chromatids, right? So they're the copies, and they're going to get separated during this mitotic phase. So there's that term centromere. There's another structure called the centrosomes, which have replicated themselves, and we can see them here and here. They contain the centrioles in there, and they begin moving to opposite poles. So that's another key event in early prophase. They're also going to have microtubules growing out of there that we're going to refer to as the mitotic spindle. And that mitotic spindle is going to be, again, it's microtubules, and these are going to be the structures that help pull apart the sister chromatids that we'll see later. Also growing from the centrosome are going to be microtubules that radiate outward, away from the center of the cell, and those are going to help support the entire structure. And they're called asters. So this is the asters would radiate away from the center of the cell towards the poles. And then we have late prophase, and here during this stage your chromosomes are now visible. Your nuclear envelope, the nuclear membrane, is now going to break apart and disintegrate. And then we're going to have the special microtubules from the spindle fiber. They're going to attach to the centromeres. So if I have my chromosome here, one of them, we have 46 of them. The chromosome has a centromere here, and the centromere on either side is going to be this plaque protein, which is going to be called the kinetochore. And so the spindle fiber or microtubule from the spindle fiber is going to attach one from each pole. Is going to attach, and eventually they'll help pull the sister chromatids apart. So that term is called kinetochore, and there's one on either side of the chromosome. And that's the point where the pulling will occur. There's going to be other microtubules in the spindle fiber. They're going to be called non-kinetochore microtubules, and those are going to be involved in pushing the two poles. So here's one pole right here, and here's the other pole. And you can see the non-kinetochore identified there. And sometimes they call them polar microtubules. And so those are going to be involved in helping to stretch the cell out as the sister chromatids get ready to be pulled apart. And then we're going to end up getting to a stage called metaphase once the connection. So we go back over here. The kinetochore microtubules have actually attached to our chromosomes, and they begin to pull the chromosomes, the entire two sister chromatids called a chromosome, and pulling them back and forth until they all meet up along the center. And once those chromosomes meet along the center and are precisely aligned, and we're going to call that center the metaphase plate. That's an important structure there. It's kind of like an equator. It's not a real structure. It's just a plane on which those chromosomes we see line up on. Once they're lined up in the middle, that would be metaphase. And as soon as this happens, they're under tension, and there's going to be a special enzyme that goes and dissolves protein that are holding the two sister chromatids together, and they immediately begin pulling apart. And once they pull apart, that phase is called anaphase. So you can see now in the picture here, our sister chromatids that were once together now are being pulled apart, and they're moving in opposite directions towards the poles. Okay, so it is considered the shortest phase, and here it says that the centromeres of the chromosomes split simultaneously, and then each sister chromatid is going to be separated. And once they separate, they're going to now be separate stained or colored bodies, so that we can refer to them again as chromosomes, now as individual chromosomes going towards their poles. There's going to be some motor proteins on the kinetochore that help drag these chromosomes along those microtubules. Again, the non-kinetochore microtubules are going to continue to push either pole outward to stretch the cell outward. And then the next phase here is telo or telophase, and here during telophase, your chromosomes stop. They've reached their poles on each side of that cell, and the chromosomes are going to start to decondense or uncoil, and so chromatin is going to reform again. You can see in the image here that we're starting to see, according to the way they colored the diagram here, we're starting to see the shading of purple around there again, which is supposed to represent our nuclear envelope. So the new nuclear membrane forms, nucleoli or nucleolus reappear, and the spindle fibers and microtubules begin to break down and disappear. You can also see that we have cytokinesis is already occurring. So you can see the cell pinching in the middle. That pinched area in the middle is called the cleavage furrow. And what helps do this is those cytoskeletal elements, the microfilaments that you see there. Those are those microfilaments that are made of the protein called actin. And so the actin begins to pull and constrict in the middle to pinch the cell off into two. And this is cytokinesis, and it starts to begin as early as anaphase. Even though we didn't see that here in this image of anaphase, it can begin as early as anaphase and then continue through telophase, and then we finally have two new cells. And so there's your cytokinesis, which was mentioned already. There's that contractile ring made of those actin filaments. There's the term actin filaments, creating that cleavage furrow, which is where the cell is pinching in the middle. And eventually you get two daughter cells, and they'll be exact copies of each other. There's an animation here on this one, and there's some animations that I have linked on the weekly documents. Make sure you check them out, and we'll probably view those during our class time together. So I'm going to go ahead and skip that animation there. Now, to be sure, there does have to be control over cell division. We can't have cells dividing constantly all the time. Otherwise, that would be like what cancer is, out of control. So there are some go and stop signals that the cell responds to, stimuli. Go signals might include the size of the cell. If the cell hits a certain critical surface volume, surface area to volume ratio, then the cell's gotten too big and it's hard for it to exchange materials with its surroundings, so the cell may be stimulated to divide. There are also some chemicals like growth factors and hormones that may stimulate the cell to begin the S phase, the synthesis phase, and then begin to go through division. There are also some signals that tell the cell that it should not bother trying to divide, and one might be the availability of space. So normal cells will stop dividing when they come in contact with other cells, and that is referred to as contact inhibition, which means when they run into another cell, that would be a stimulus to tell them, don't divide any further. We don't need any more growth by cell division here. There are some proteins in the cell that are involved in this regulation of whether or not a cell is going to enter into S phase and then go into mitosis, and they're called cyclins. And they're called that because they are observed to cycle. They increase just before cell division occurs, and then after cell division, then the cyclins go away or decrease in concentration within the cell. So they seem to be involved in helping to regulate. It had been discovered from some research that there was other protein involved, and this one is CDKs or cyclin-dependent kinases, which have to combine with the cyclins. So these cyclin-dependent kinases, they depend on the cyclins, and the cyclin-dependent complex. When these two bind together, that activates the complex, and that complex now then goes and activates other enzymes that go into a cascade or a series of reactions that promote the cell to go and divide. After cell division, again, the cyclins are destroyed, broken down, and the process stops, at least for the time being, while the cell is just going through normal growth and activity during interphase, or G1, the gap 1 stage. So There are some checkpoints that the cell makes sure that things are going smoothly. So these are going to be we want to make sure, for example, that DNA was copied correctly. For example, so the G1 checkpoint is the main restriction point, and that one, if the cell is stimulated to divide and everything is good to go, and we pass this checkpoint, we're now committing to division because we're going to make copies of DNA. That's a big commitment because a lot of resources are needed by the cell to make copies of DNA. So a major commitment is made right there at the G1 checkpoint. If the cell doesn't pass that checkpoint, then the cell stays in G0, which would be true, say, of neurons, which are cells of the nervous system, and cardiac muscle cells, for example. You see there's another checkpoint right here, the G2 M checkpoint, which is right between G2 and mitotic phase. This one would check to make sure if the DNA is copied correctly and everything is good. At that point, we can progress to dividing the cell. So here's a question here. During which phase of the cell's life cycle is DNA replicated? So in section 3.11, we're going to be looking at protein synthesis, and your learning outcomes are going to be to define gene and genetic code and explain the function of a gene, and then name the two phases of protein synthesis and describe the role of DNA, mRNA, tRNA, and rRNA in each phase, and then contrast triplets, codons, and anticodons. So regarding protein synthesis, your DNA is the master blueprint for holding all of the codes needed to make protein. So for basically giving us the order in which amino acids will be put together to make our polypeptide. A segment of DNA that holds the code for one polypeptide is going to be referred to as a gene. So the definition of gene is a segment of DNA that codes for your protein or your polypeptide. So the code is going to be determined by the specific order of those nitrogen bases. Remember, there's adenine, guanine, thymine, and cytosine, and it's the order of those that determines. Now, the code consists of 3 sequential bases, which we're going to call a triplet. So on DNA, every 3 bases that are part of coding for a proton, every 3 is going to be referred to as a triplet. For example, the triplet GGC is going to code for an amino acid called proline, which is one of the 20 amino acids, whereas the code GCC would code for arginine. So this code will give the instructions for providing those amino acids, and then there are other codes for other amino acids. So each triplet specifies a code then for a particular amino acid. Now, genes are going to be composed of, with those segments of DNA, they're going to be composed of exons and introns. So exons are going to be the part of the gene that actually codes for amino acids, and introns are going to be non-coding segments that are interspersed among the exons. So if we look at the picture here, here's your DNA. And so your DNA would have segments of introns, which we might refer to as interfering segments, whereas the exons would be expressed segments, ones that actually are made, going to be used to make protein. And so we're going to take our DNA and we're going to use our DNA to make a messenger RNA that will take that message to your ribosomes. And your final product RNA would have those introns removed from there. We'll see how that works a little bit later in this video. So what is the role of RNA? RNA, it's the go-between molecule that links DNA to proteins. And so basically RNA copies the code from DNA, and this has to happen in the nucleus because that's where you find your DNA. And it's going to carry it to the ribosome. So generally, the following pattern results. Your gene in DNA is going, the sequence of those bases in DNA is going to be copied into a sequence on your RNA. And in this case, if it's carrying the actual message for a polypeptide, we'll call it mRNA for messenger. And then that goes to the ribosome to make your protein. And the ribosomes are found in the cytoplasm. Now, all the NRA that we're going to be dealing with is formed in the nucleus. And RNA, you might recall from chapter 2, does differ from DNA. One of the ways it differs is we have a base uracil instead of thymine. Thymine is found only in DNA, whereas uracil is in RNA. You might also recall that RNA has ribose sugar instead of deoxyribose for their nucleotides. And RNA is also single-stranded. There's going to be 3 types of RNA. There is mRNA, which stands for messenger RNA. There's ribosomal RNA, which is rRNA, and transfer RNA, which is for tRNA, or abbreviated tRNA. So what is the different RNAs? We'll start with messenger RNA. It's a single-stranded piece of DNA, and it basically is going to take the code from the DNA template and copy it onto for the mRNA. So basically, when the DNA is pulled apart, we're going to read a template strand. So looking at the image here at the top right, we pull apart our DNA, and one of the strands is going to be used to make your mRNA, which is this yellow molecule here. So whatever the sequence is for this template strand of DNA, the complementary bases will be placed on that mRNA. So basically, the mRNA would be carrying that code. Since this is the same language as nucleic acid chemical language, biochemical language, we are basically making what's called a transcript of that message in this mRNA. So we're going to call this process transcription. Now, the 3-letter codes that were called triplets on DNA are going to be complementary to 3-letter codes on mRNA, but we're going to call those codes codons. So the triplet is on your DNA. The triplet makes the codon on mRNA, so that's the key term there. So where are codons found? They're found on the messenger RNA. Now we look at ribosomal RNA, and you might recall that ribosomes are produced in the nucleolus of the nucleus. And basically the ribosomes are made of, at least in part, by ribosomal RNA. There's also other proteins that help produce the ribosome structure, but they do have rRNA, which is ribosomal RNA. And along with tRNA, which we briefly mentioned a while ago, it's going to help to translate the message from the messenger RNA into a different chemical language, polypeptide or protein language. So we look here at our, I went that far there. So looking at transfer RNA, the tRNAs, those are going to be carrying amino acids to the ribosome. And they're going to have special areas. They're going to have their own triplet code. We're going to call that the anticodon, which would be complementary to the codon on the mRNA. So DNA has the triplet, RNA carries the codon, and tRNA matches with the anticodon. So each tRNA with its specific anticodon will be carrying a very specific amino acid. So there's a specific codon for the anticodon, and that tRNA has that amino acid being carried with it. So the anticodon will complementary pair up with the codon on your mRNA, and the ribosome assists with this process as we bring in amino acids to make our polypeptide. Now this process that tRNA is involved is called translation. So looking over here, we covered transcription, where we take our template strand on DNA, make our mRNA. Then the mRNA passes into the cytoplasm and then finds our ribosomal RNA in our ribosome. And so then you're going to have your tRNAs here that are going to be bringing in, based on the codons, the 3-letter sequences that are on the mRNA, the tRNA will match up its anticodon, bringing in the correct amino acids. And so we'll have our polypeptide chain growing as we continue to read along the mRNA. Ultimately, once we get to a stop on this process of translation here, the ribosome, mRNA, and the polypeptide break away from each other, and then the polypeptide will then go into a folding to go into its functional structure. So overall, we know that, as we've discussed, we have 2 processes. DNA, when we use it to make mRNA, that process is going to be called transcription. since we're in the same chemical language. And then when we want to change chemical language, going from our nucleic acid language to protein, at the ribosome, and this is in the cytoplasm, that's going to be called translation, because we're changing chemistry, we're changing the chemical language. So translation, your mRNA is going to be decoded to make your polypeptides, and that occurs at the ribosome. So looking at transcription, it's a process with a few details you should know. This is going to be the process of transferring the code held in DNA in a gene and the base sequence to complementary base sequence in mRNA. Okay, so we're going in the diagram here, we're talking about transcription. We've got to separate the two strands of DNA, use a template strand to make our mRNA. Now, what's going to be required for this process are transcription factors, which are basically proteins. And they're going to help activate transcription by the following. First, they're going to loosen the histones from the DNA in that area that we're going to be transcribing. That way we can expose the DNA. And then the transcription factors also help in binding to a special sequence of the gene to be transcribed. And that special sequence is called a promoter, which is the starting point. So this is where we'll actually begin the process of transcription. Now this is going to occur on the template strand. So remember, when you pull apart the DNA, there's two strands, but the strand that's going to be used is called the template strand. Now, mediating the binding of the enzyme that's going to go to work is the other job that these transcription factors do. So they assist, when we say mediating the binding of this enzyme called RNA polymerase. That's what the factors also help do. And so the RNA polymerase is kind of like DNA polymerase, except the RNA polymerase is going to be able to now an mRNA molecule. There's going to be three main phases in this process. There's going to be initiation to start, then elongation as we build our big long mRNA strand, and then termination. So let's take a look at it through the diagram here. And so in initiation, with the help of the proteins, where our RNA polymerase will find the promoter region, which is going to be a segment there. And remember, one strand is the coding, but the one that's going to actually be used to make our mRNA is called the template strand. And so when our RNA polymerase binds, it's going to pull apart the two strands, unwind and unzip, and we're going to begin making our RNA. And we're always going to be adding to the 3' end. So we can see our RNA polymerase is running in this direction. This is our RNA polymerase. And so we're going to be running along there, and we're going to be using the template strand. In this diagram, it's the lower strand there. As the RNA polymerase continues to add more nucleotides to the 3' end of this growing mRNA strand, that process is called elongation. And we zoom in here to see it more closely. At any point in time during this process, we're adding, you can see those nucleotides being brought in. They're coming in and they're going to match the complementary bases from the DNA. So if DNA has a G here, then we would be bringing in a C or a cytosine. Remember, we don't have U's. We don't have thymine. So if this was an A right here in the next position, then what we would be getting is uracil coming in on our new nucleotide. And so the RNA polymerase is going to go and continue to read the template strand. And at any given point in time, you have this sort of a hybrid where you have an RNA and a DNA strand temporarily bound. So anywhere from 12, 16 to 18 bases are joined around there. I think the number the slide before gives 12. And here at any given point in time, the RNA polymerase is pulled apart somewhere around 18 or 20 bases as we're going. And then behind there, the DNA molecule winds back to its normal structure. Now, the termination is going to happen when we get to our termination signal here at the end. And when we do, the RNA polymerase falls off and you have your mRNA strand falling off. So here's your now mRNA strand, and this again has occurred in the nucleus where the DNA is found. So once we finished, we actually don't have a mature RNA, mRNA. We have pre-mRNA, so it needs to be processed, and that's where we're going to go and cut out the introns, right? So we have to go and do that to the pre-mRNA, and what's going to help do that are going to be complex structures that are made with small nuclear RNAs and protein, and other proteins are going to come together to form our spliceosome here, which is going to get our two exons. Here's exon 1 and exon 2, and we need to cut out that intron. So the spliceosome, which is this complex body right here, will help form a loop of the intron, cut that loop out, and then splice together the two ends so that now exon 1 and exon 2 are together. And so when we finish that and make our mature mRNA, that mature mRNA will go out into the cytoplasm to begin the translation process. So for translation, translation is where we're going to go from nucleic acids language to protein language with our amino acid sequences. So the process is going to involve using mRNA. We're going to need a genetic code, the code that every 3 letters is going to be a code for amino acid. We're going to need our transfer RNAs and our ribosomes. We're going to need those events that occur during translation. And sometimes if we're going to make protein for excretion, for example, this will be occurring in the rough endoplasmic reticulum. So looking at the genetic code, we see that every 3 letters from DNA is your triplet. That triplet becomes a 3-letter sequence on your mRNA, which is our codon. And so those codons are 3 letters, and if we have 4 letters in our genetic alphabet, there's the A, the T or U in RNA, the C and the G, 4 letters possible. And since we're going to be reading the codons off mRNAs, A, U, C, and G instead, there's 4 possibilities for each of the 3 positions. So we have 4 possibilities for the first base. The next base on the mRNA is 4 more possibilities, an A, U, C, or G. And in the last position on the 3-letter codon is also 4 possibilities. That gives us 64 possible combinations, and we see them here in this chart. Okay, so in the chart right here is our genetic code, and you can see the 3 letters. The first letter is on the left side, and then we can find our second letter at the top of the chart, and then the third letter or third base on the right side overall. And if we look, there's one codon that's always for start. So AUG will always start, and it's always going to start with amino acid MET, which stands for methionine. And because we only have 20 amino acids, there's some redundancy in here. So when you look, for example, there's 1, 2, 3, 4, 5, 6 codons for leucine, and isoleucine has 3, and then valine has, it looks like it has 4. So there's more than enough codes where we have some amino acids have more than 1 code. So that's the case. Now 3 of the codes are going to be to stop the process, so they're referred to as stop codons. You can see them right here in the chart here. UAA, UGA, and UAG would tell the ribosome and the entire process to stop the translation so that the polypeptide is complete. So looking at the role of tRNA, which is the transfer RNA, the tRNA, and there's a model of it on the right, the picture on the right here, that tRNA is going to bind a specific amino acid at one end, the stem. So here's your stem right here, and there's an amino acid, isoleucine. Now, once the amino acid is loaded onto the tRNA, the molecule is now called an amino acyl tRNA. So here we have our amino acyl tRNA. It's loaded or charged with its amino acid. It's also going to have an anticodon on the other end, or the head end right there, and that anticodon that's right here, in this case UAU, is complementary to the codon that codes for this amino acid. So the codon, the actual match on the mRNA, the messenger RNA, would be AUA. and AUA actually codes for isoleucine. So this particular anticodon matches with that specific amino acid, right? So that's what it's trying to say here. If there was an error in the original notes that said methionine, the actual code that we see here is an anticodon for isoleucine. So I made that correction just to be consistent with our genetic code here. So the anticodon of tRNA is going to bind only with a codon on the mRNA that's complementary to it. So, for example, the codon AUA is only for a tRNA with the anticodon UAU for the other one. So you can see that one was right here, AUA, and this is our mRNA right there, and you can see that was an isoleucine. The next codon that's on there must be coding for this other amino acid, proline. And then we see another codon here, CUA, and there's the anticodon, and it's bringing in an amino acid called leucine. So CUA should be the codon for leucine. Let's see if that's the case here. CUA, and it is leucine, so the image seems to be correct as far as the amino acid that we're bringing in right now, according to that codon matching those anticodon. That's a match right there. Okay, so what does the ribosome do? And which again has the ribosomal RNA with the protein in there, but what does the ribosome do? The rRNA is involved in coordinating this coupling between the mRNA, its codon, and the tRNA with the anticodon. So it coordinates the matching up of those two so that we can bring in the correct amino acids. So the ribosomes themselves are going to contain one binding site for the mRNA. So it hangs on to the mRNA that we see here. That's your mRNA in the diagram. And then it's going to have three binding sites for tRNAs. One is the amino acyl site, which is the A in the diagram here on the right. And then the other is the peptidyl site, which is the P, the middle site. And it's called that because that's where we have our growing polypeptide chain. So P for peptidyl site. The A, by the way, for the amino acyl tRNA coming in. And then E, which is the last one there, is going to be the exit site. And that's where a tRNA that's already given up its amino acid to the growing chain is going to exit out from there. So E for exit. So the sequence of translation or the events are also in three phases. The first one is initiation, and this is kind of like the way transcription was. Same names. The second one is elongation, and the third is termination. So you have the same names. The events are different though because it's a different process. Now all these events are going to require some energy, ATP, protein factors, and enzymes. So let's take a look at initiation. So initiation is going to happen here, and I'm just going to go ahead and describe it. It's you can read about it here, and you can read about it here, but let's take a look at what's happening. The initiation is going to happen when we get our start codon, which is AUG. Go back there. AUG, and you're going to get your small ribosomal subunit. So this one is actually smaller. This one's supposed to be larger. The picture doesn't really show that. They kind of look roughly about the same size, but the larger subunit is the other side. So the small subunit is going to bind with the mRNA, which is this molecule right here, the one that's carrying the code from the DNA. And it's going to bind right around the area where the AUG or the start codon is, right there. And the first amino acyl tRNA is going to come in, and it's going to be the one carrying that anticodon, which will always carry the methionine amino acid. So we're always going to start our polypeptide with methionine. Okay, and once those three molecules bind, we have our small subunit, we have your mRNA, you have your tRNA carrying methionine, then the large subunit binds. And once all of those components are together, all three components, your small unit, large unit, subunit, your mRNA, and your tRNA, we've now initiated. So the process is now started. The next is now elongation, and there's three main steps. The first one is code recognition, and then peptide bond formation when the amino acids are joined, and then translocation, which means to move. You're going to be moving down, translocating, you're moving. So we're going to be moving along the mRNA molecule that has all the codes on it. Okay, so let's take a look at those 3 stages. So for elongation, part 2A, or the first step there, we're going to have code recognition. So we already have the we're already going to have a charged tRNA in there. We've already started we've been elongating already. We already started with our methionine, and we've already added several amino acids. And every time we do, we're moving or translocating along there. But here now, we are going to be code recognizing at the A site. So this A site has a codon, and we're now going to recognize and bring in the correct tRNA with its amino acid, and we bring it in, and the anticodon has to match the codon. So we're going to bring into the A site, and that's code recognition. Once code recognition has occurred and your tRNA comes in and binds in the A site there, and codon and anticodon match, we're now going to have the second step, which is peptide bond formation. So now we're going to actually join that new amino acid to the growing chain of amino acids. There goes the leucine being added. What the chain was already there is going to be added to the end of that leucine is going to be added to the end of that chain, of that growing polypeptide chain. And then once that occurs, then we're going to translocate. So we're going to move the entire structure, a growing peptide, we're going to move all of this down 3 bases to the next codon. And when we do that, the former tRNA that was in the P site moves to the E site, and it's now empty. It's lost its amino acid, so it gets released, it gets ejected, it exits, which is why we call it the E site. And so now that we've gotten here, we're ready to start again with code recognition. So we go to the next code, and the next amino acid will come in to that. So every time this process, we're repeating this cycle, and we'll continue to repeat this with a new amino acid coming into the A site with its tRNA, the peptide bond formation, and then ejection of the used up tRNA, and then we move along again. So the process repeats over and over again until we get to our stop codon. And so that elongation is all described here. And there is cases where we can be actually transcribing several polypeptides at the same time, and that's modeled here in the diagram below here. It's called polyribosome here, and so you can see that we can have initiation start several times along the chain, and as we move the mRNA through the ribosomes, all the ribosomes, we have various stages of completion of the polypeptide. So here comes a new ribosome binding at the start codon there. We have another ribosome that's a little further along, so you already have the growing ribosome, and then another one also translating there. And as we move further along, these ribosomes have been on the mRNA longer. They're making, overall, you have a longer polypeptide, and then you eventually get to the stop codon, and we're breaking away from there. So that's called polyribosome, and there's an electron micrograph showing all of the ribosomes on the mRNA. So they're all translating at the same time. So polyribosome then would be making multiple copies of that same protein at the same time. That just, again, the ribosomes would be at different stages as they move along this mRNA. And then we're going to have termination. And remember, there was 3 stop codons. So there was UGA, UAA, and UAG. And what's going to happen there is on those stop codons, there is no tRNA for that. So what's going to happen there is instead, there is a protein called release factor that recognizes those codons. So the protein comes in there, not a tRNA, not an amino acyl tRNA comes in, but a release factor comes in. And when it binds to that spot, that causes the whole complex structure to break away. So the small subunit, you can see over here, the small subunit comes off, the polypeptide chain is released, and the large subunit is released. And so now your polypeptide has to be processed further and possibly folded to become its functional protein. That entire process And those images are in this diagram, so it's a nice summary. I encourage you to read over it and try to explain it, maybe sketch it to help you learn the process. Now, what about when translation occurs on the rough endoplasmic reticulum, which is what we would be doing if we wanted to keep proteins separate from the rest of the cytoplasm. For example, if we're making a hormone like insulin and we want it to be secreted outside of the cell, we might produce that protein within the rough ER. So how that happens is going to be shown in the following diagram, but there is going to be a little bit extra here. There's going to be a short amino acid segment, and this is going to be called the endoplasmic reticulum signal sequence. And it's going to be present on the growing polypeptide chain, and it's going to signal for docking on the rough ER. So there's going to be a signal recognition particle on the endoplasmic reticulum, and that directs the mRNA and the ribosome to dock onto the endoplasmic reticulum. Now, once the complex docks, the polypeptide then is going to enter the endoplasmic reticulum. So the peptide will continue to be made, but the peptide will be placed inside of the endoplasmic reticulum. To be clear here, translation actually starts in the cytosol on a free ribosome, but then that particular protein has that small short segment called the ER signal sequence, and that then gets recognized, and that's where it now docks onto the endoplasmic reticulum to start translating the polypeptide to go into the endoplasmic reticulum. So now when the polypeptide is going into the endoplasmic reticulum, sugar groups might be added to the protein and the shape might be altered. And then the protein is going to be enclosed in a vesicle, and it'll eventually be transported to the Golgi apparatus for further processing. So here's a diagram showing the process here. So again, your ribosome is already going to bind to your mRNA as we described before, but the difference here is that the sequence that's created is going to have this signal on it. So there's this little signal sequence, looks like this is off. The arrow is supposed to be pointing to that little bracket, should be pointing to what I just circled here. So that's your little signal sequence in green there, and it recognizes this SRP, or signal recognition particle, which is that black structure there, which was already on the endoplasmic reticulum. And so that recognition causes this structure to bind here to the endoplasmic reticulum. And now once that occurs, then the polypeptide will start to fall inside of the endoplasmic reticulum. So you can see that occurring right there. And as we continue to translate the code from the mRNA and have our polypeptide growing in length, we continue the process, and sugar molecules may be added to the protein. The protein may be folded up, but eventually it's going to end up being placed into a transport vesicle that will go find its way to your Golgi apparatus for further modifications and repackaging into perhaps a secretory vesicle. So this is how our rough ER processing of proteins works. And again, might be how something like insulin is produced. This might be also how a lysosome is produced. So what you end up with after going through the Golgi is a vesicle that stays inside and does intracellular digestion when it comes to a lysosome. So our summary from DNA to proteins. Complementary base pairing is going to direct the transfer of genetic information from DNA into the sequence of amino acids for our protein. Remember that DNA has the triplets, and those triplets are then coded on mRNA, and we're going to call that three-letter sequence on mRNA, we're going to call those codons. The mRNA codons are then going to be base paired with the tRNA anticodons to ensure that the correct amino acids are brought in so we get the right sequence. The anticodon sequence on the tRNA then is going to actually look identical to the DNA sequence, except that uracil is substituted by thiamine. So let's take a look at the overall summary without showing all the other structures in there. So you start off with your DNA. And then your DNA is going to have a segment. So there's different segments. You might have gene 1, gene 2, gene 4, whatever gene is going to be basically transcribed and then translated. So here it looks like they got a little segment out of gene 1 just so that we can focus in on a small piece to help understand the overall larger process. So this would be your sequence in the DNA right here, okay? And so those would be your triplets. So this is triplet 1, triplet 2, and so on. And then those the triplets then are going to be transcribed onto mRNA. So this is our mRNA. And so transcription would have to occur here. And this occurs in the nucleus. So now every 3 letters is now called a codon. So this is your codons. Our start codon would be AUG. The next codon matching the triplet from DNA would be CCA, and so on. And then, and that does match, G matches with C, and so on. Now remember, you can't have T's in mRNA, so the A would have to actually give us a U for the mRNA. Now the anticodons, which are found on the tRNAs, and they're carrying very specific amino acids, are going to be complementary to your codons. And so A would match with U, U with A, and G with C overall. So here's your anticodons on your tRNA. And remember, this process going from mRNA to a protein, all of these would be involved in the process of translation occurring at the ribosome. Right? So the slide earlier said that the anticodon should match our triplets in our DNA. Let's see if that's true. Remember, you have to have an exception. There is no T's in RNA, so we would have a U instead. So the U would be a T in DNA. So that matches. The A on the anticodon would match, and it does with the A, and the C matches with the C over here. So that's what that last statement was saying on the prior slide. So overall, this is summarizing everything. You get your sequence in your DNA, which is this one here, and every 3 letters is your triplet. Those are going to correspond by pairing rules, by a complementary basis, to your sequence in your mRNA. So your code is being carried here in your tRNA, and then your in your mRNA, and your tRNAs are going to be bringing in those amino acids based on the sequence of every triplet. So the codon CCA is going to guide and say we need to bring in this amino acid called proline. And that's according to the anticodon matching the codons. So here's just showing a few of the 3 amino acids that would be brought in. So remember, every 3 letters is for an amino acid. So if I wanted to make a polypeptide that was 100 amino acids long, that would require 300 bases to do that. So because every amino acid requires 3 letters. So 100 amino acids would require 100 triplets, codons, or anticodons would require basically 100 codons, and every codon has 3 letters. So that would be 300 bases altogether. So there are other types of RNA. I think here at the top it says roles of other DNA, but we'll be talking about RNA, so there should be roles of other RNA. So DNA can code for other types of RNA. There's microRNA or miRNA. These small RNAs that combine to and silence mRNA, and that would be their roles. And then riboswitches. These are folded RNAs that act as switches that can turn protein synthesis on or off in response to certain environmental conditions. And then there's small interfering RNAs or siRNAs, and these are similar to microRNAs as mentioned right above, but can also be made to silence mRNA from disease-causing sources like viruses. So these might play a role in defense against pathogens. So the main function of DNA is to dictate the production of, is it A, protein, B, carbohydrate, C, lipid, or D, nucleic acid? So in section 312, titled Apoptosis, Autophagy, and Proteasomes, here in this section, your learning outcomes are going to be to distinguish between autophagy, apoptosis and indicate their importance, and then describe the importance of ubiquitin in dependent degradation in soluble protein. So what is the role of this protein, ubiquitin, in breaking down certain proteins in the cell? So what is autophagy or autophagy? When cells have become old or damaged, they need to be taken out by your system, and so this is called autophagy or self-eating, where we have a process of disposing of non-functional organelles and sweeping up cytoplasmic bits by forming autophagosomes, which can then be degraded by lysosomes. So you create vesicles that lysosomes can go and break down, say, worn out parts within the cell. And then if we need to get rid of proteins that are no longer needed or they're damaged, then we have a way of doing that. So that's going to be done by marking those proteins for destruction with a protein called ubiquitin. And once that's happened, then proteasomes, which are protein bodies, are going to help disassemble the ubiquitin tagged proteins. And when they do that, basically they're breaking down all of that protein into amino acids. And then there's apoptosis, which is also known as programmed cell death. And this is going to happen in certain cells to self-destruct. This happens as a normal part of development. You have like webbing between your fingers early in development, and those cells go through apoptosis. Another example might be the tail on a tadpole of a frog. During their development to adult, the cells that make up the tail self-destruct. Other cases where this can happen is in cancer cells, so a protective mechanism would cause the cells to go through apoptosis. Also, infected cells or just old cells would go through this process. Now, the process does begin with the mitochondrial membranes. The membranes in the mitochondria are going to leak chemicals, and these are going to activate enzymes called caspases, and these caspases cause the degradation of DNA in the cytoskeleton. That is going to lead to the cell death. The dead cell then shrinks and then is going to be phagocytized by macrophages. So our macrophages will come in and eat them up and digest down these cells. For developmental aspects of cells, all the cells in your body, all trillions of them, have the same DNA, but not all cells are identical. We have like over 200 different cell types in there. But chemical signals in the embryo are going to channel cells into specific pathways for development. So some cells are going to go on to become connective tissues. Some cells are going to go on to become muscle tissue, and so on. And this is accomplished by turning some genes on and others off. So we're talking about switching on a certain set of genes to cause these changes in the cells to occur. So the development of these features by these specific cells is referred to as cell differentiation. So we start off as one cell called the zygote, and eventually we end up with 250 different cell types because of differentiation. Now, this little section here called cell destruction and modified rates of cell division here. Organs are well formed and functional before birth. But in order to grow, we grow by cell division. So cell division, for example, might be needed to replace cells that are like epithelial cells that become damaged or fall or worn off. We might need that cell division to repair wounds. And so when we have this kind of growth or repair by cell division, that process is called hyperplasia. And that's hyperplasia is an increase in cell numbers. So by cell division. There's another kind of growth which muscle cells and adipocytes do, fat cells, called hypertrophy. I don't know why this pen does that every now and then, but let me try to rewrite that again. The process is called hypertrophy, and here the cells don't divide, they enlarge. So muscle cells, muscle tissue, and fat adipose tissue would grow by hypertrophy, which is just the cells themselves getting larger. There's another related term or opposite to hypertrophy is atrophy, and here The cells decrease in size because of loss of stimulation. So while muscle can hypertrophy from exercising with weights, you can lose that muscle, those muscle cells, through shrinkage, and that's called atrophy. Now, what about aging? How do cells age? There are several ideas or theories, hypotheses about this. One is wear and tear theory, and just a lifetime of chemical insults and free radicals have a cumulative effect of being destructive within cells. The other one is mitochondrial theory, and here free radicals, which are very highly reactive compounds in the mitochondria, are going to lower energy production, which would not be good for your metabolism. And then immune system disorders here, autoimmune responses, as well as progressive weakening of the immune system overall. Autoimmune responses when your immune system begins to overreact and start attacking your own body cells. And then there's genetic theory, and here basically mitosis is going to stop, and it's programmed into the genes. So there are structures called telomeres, which are at the ends of your DNA molecules. Your DNA molecules are linear, and every time they divide, a little piece of the telomere falls off. And they know the mechanism for this and how it occurs. So basically, you're losing the little ends of your DNA that help stabilize the molecule. And once you lose the entire telomere, that's when the cell stops dividing. So that line of cells dies out. So due to that telomere shortening, there is an enzyme called telomerase that is going to become active in germ cells. Your germ cells are the cells that would produce sperm and egg cells. So basically we reset that sort of hourglass. It's kind of like, we compare it to like an hourglass. When you turn the hourglass over and the sand runs out, kind of resets the clock on there with this enzyme that becomes activated during in the cells that are going to produce sperm and egg cells. This telomerase, for whatever reason, does become reactivated in cancer cells. So when cancer cells form, the enzyme is not normally present in our normal cells of our body. But if a cancer cell forms, then the telomerase gene kicks back in for whatever reason and basically reestablishes these telomeres. So that makes cancer cells sort of immortal. And they've had some cancer cells that they've been studying since like 1950. They're still growing in culture, and they basically have indefinitely a long lifetime on. They're capable of dividing forever, as long as there's the right conditions for cell growth and division. The telomeres will never run out on those. Here's a homeostatic imbalance, 3.4, a clinical situation, a condition called progeria. It's a rare disease, and it mimics aging, and it's caused by a defective progerin protein in the nuclear lamina that results in an unstable abnormal nucleus. The disease would appear by age 2. And children with the disease display slow growth, thinning hair, brittle bones, arthritis, severe cardiovascular disease, and death, usually by age 20. Scientists have found a drug that does stimulate autophagy that can help cells clear out that progerin. And I don't know what effect that this has, maybe perhaps slowing the progression of that disease. So looking at practice question here, what is autophagy? Is it A, digesting bits of cytoplasm and excess organelles? Is it B, degrading excess extracellular materials? C, degrading excess soluble cytosolic proteins? Or is it D, removing excess cells during development? And then cell differentiation occurs when early embryonic cells, is it A, cut out part of the DNA and destroy it? Is it B, switch off certain unneeded genes? C, separate and form a different parts of the body? Or D, die in the process of apoptosis?